afternoon. And you hear that lady clapping here, and that's the lady that just would take the scene for 1 minute or two for a small announcement.
Oh, yeah. So we're going to have an air guitar competition later on. So anybody who wants to join to practice the air guitar, let me know. Thanks.
Okay, great. Thanks, Sarah, for making this meeting even more beautiful. And with this, it's an absolute honor and pleasure for me now to introduce the first speaker to this session, which is, I would even say, the father of T7. I think there is, no, he says no, but I think there's really many of our old and beloved companions here in this audience who have been here for long, and each of them has a phage with that he or she is associated, and Ian definitely is associated with T7, and we're looking very forward to your talk, Ian, a talk about the complex pathway of T7 adsorption to lipopolysaccharide.
Maybe I should give a little preface here. So T7 was first isolated and defined as such in 1945 by Demarek at Cold Spring Harbor. A few years before then, Max Delbruck had isolated a phage which he called Delta. And Delta and T7, as far as anybody could tell in 1945, were identical. But work had gone on with both phages, and Demarek and Delbruck agreed that one of the two stocks, in this case it was going to be Delta, should be destroyed. So there was only going to be one T7. Delbruck could never remember whether he ever did that or not. But it turns out that it probably didn't matter. And in fact, T7 was probably isolated back in 1921 by Felix Durrell, but not T7 itself, but a T7 wannabe that actually grew on Yersinia pestis. And that phage was actually used and became the story of Arrowsmith for phage therapy, book that was published in 1924 or so. But coming more recently, so the father of T7 is really Bill Studier, who retired just a few years ago. And I guess I'm, in a way, his scientific son. He's been a great help to me over the years. I have many of his mutants, which is fortunate because his mutant collection has now gone. So what I have is what survives. So what I'm going to tell you about today is a little bit about adsorption, which has been driving me crazy for quite a while on terms of trying to understand it in T7. So T7 itself binds to LPS. We know in E. coli B strains the exact residue on the LPS that it binds to, and T7 also grows on coli K12 and E. coli C with a slightly different LPS structure. We don't know what residues it binds to directly. But if we go back to early phage biology, this work was done by Gunther Stent, Ellie Wollman, and they came up with this scheme originally that you had phage, you mixed them with cells, you got reversible adsorption, and this is a functional definition. This simply means it's not a mechanistic, it's purely functional. It means you can go back and isolate phage from that complex which have maintained infectivity. And you go from this step down to irreversible adsorption, and then you get infection and phage growth. That was their proposal. And it turned out that Luria, another guy you've probably heard of, objected to this. And what he pointed out was that there was no evidence in any system he was aware of that you went from reversible adsorption to irreversible adsorption. And in the one case that I'm aware of off the top of my head at the moment, where this has ever been looked at, is in SPP1, where Caterina Baptista showed that this was not a precursor to this. You had to desorb and then go down to an irreversible adsorbed state. So these simple ideas that people have had over the years are not necessarily really reflecting the truth. So in the case of T7, this is obviously a reconstruction of T7 that was done in Jun Lu's lab a number of years ago in a collaboration. And we could show, this is tomography, we could show that this is bound to a cell, or more specifically a mini cell, and this is still at a stage of reversible adsorption. We can dissociate that phage and get back infected particles. The irreversible step, which is another functional definition, it's not a mechanistic definition. The functional definition is that proteins that you can just about see them up here, they're up in the phage head, they're called the internal core, have come out from the phage particle and made this extended tail across the cell envelope. Phage has lost a lot of its proteins. It's actually lost about 2 megadaltons of protein, and it's clearly lost all infectivity unless this particular cell can be the product. We've been trying to understand how T7 adsorbs to cells for a number of years. Here's just a schematic of the particle. It's obviously the tail machine that makes the first specific contacts with a cell surface. Many of the times, the initial interaction between any phage and a cell will be through the head, because that's just the way that random forces, Brownian motion took it, but we'll consider those to be non-specific. Everything is going to be, that's specific is going to be down here in this tail complex, which in the case of T7 consists of the tail fibers, the side tail fibers here, a protein, which I'll just call GP7.3, and I'll say much more about that, and a pair of proteins, one of which is at the tip, or the nozzle, or the knob of the tail, which is GP12. GP11 is an adapter protein that joins the portal protein to the rest of the tail. So this is where we would expect to find mutants that are involved in the adsorption process. And so we've been trying to isolate lots of different mutants in affecting these proteins and measure adsorption kinetics, measure host range, because we're trying to understand not only what the proteins recognize, but the energetics of how they actually get on to infect cells. And so the logic is that if we can bypass some mutants that we construct that have known adsorption defects, we could isolate mutants that expand the host range. So in the case of T7, I'll mention about growth on Yersinia pestis, or reduce the activation energy for infection. So we can get T7 normally grows normally down to about 16, 15 degrees Celsius. We've got mutants that grow at 6 degrees. And almost by definition, since these are making plaques, they have to have a reduced activation energy of infection. Don't understand any of the mutants that we've got in that sense as yet. We haven't given up yet. Okay, so here are the 3 prime candidates here that I mentioned before. So we've got the tail machine. This is wild-type T7. And in a standard adsorption assay, in the space of about 30 seconds, a good stock of T7 has absorbed 95%. If we take off the tail completely, this is the residual adsorption. This is done by radioactive label, so we're not obviously measuring anything important here. If we get rid of the tail fibers, then we find no adsorption to speak of. And if we get rid of the 7.3 protein, again, there's no adsorption. You can extend these adsorption assays out for an hour or so. This guy is obviously all done with. Okay, so there's one complication to this, and that is here, that particles that are genetically lacking the tail fiber gene also lack GP7.3. But it turns out that can be very misleading. They're actually assembled in the presence of the protein. It just isn't there in the particle itself. So some of the defect associated with tail fibers adsorption may be because we're lacking 7.3 in the particle. Okay, so switch just for a second into a different phage. This could be a Yersinia pestis phage called Phi-A1122. It's almost identical to T7. Its genome is synthetic. All the genes are there. And this phage is used diagnostically by the CDC for detecting Yersinia pestis in the environment. So it doesn't grow on E. coli, it doesn't grow on any other Yersinia strain, but you can isolate mutants, and if you isolate mutants that grow on E. coli, every one that we've isolated lies somewhere in the gene for the tail fiber. This is what everybody would say. Why are you bothering to tell me this? I know this. Every phage is like this. Most of the mutants are in the receptor binding domain. Not all. The most common turns out to be a tail binding domain. And from structural work, we can explain why some of the host range mutants are down in the tail binding domain and not where everybody says the mutation should be in the receptor binding domain. So this is standard story. There's nothing really new here. And in common to all other host range mutants of this sort that you select for directly, they're host range expansion. They still grow on the original host as well as on the new host. And in this particular case, they go pretty much as well, equally well on both pestis or E. coli. But just to preface, okay, so this basically says the same. We've got an isolated tail fiber. If we take the Yersinia pestis phage and we put T7 fibers on it, or we put T3 fibers on it, both of these being coliphages, we also change the host range. So that's all consistent. But if we take the Yersinia tail fibers and put them on T7, nothing happens. It still remains a coli-specific T7. So fibers are not the sole determinant of host range. In fact, won't talk about this in any detail. Essentially, all of the mutants that we've got in T7 that grow in Yersinia pestis and in coli have substitutions affecting the tail protein GP12, or there's other tail protein GP7.3. So I'll come back to the tail fibers now. So here's a fiberless phage. And if we can propagate this normally, obviously with a complementing plasmid. But if we take a stock of that phage and now plate in the absence of any plasmid, no complementation, at what appears to be less than mutational frequency, we can get plaques that now grow. And this phage will now grow in the absence of side tail fibers. Now, those of you who heard Cecile's talk yesterday or Nikolai's talk today, lambda, T5, they don't care about tail fibers, side tail fibers. This guy does. He doesn't like to grow in the absence of any, but a single mutation will allow T7 to grow. And we've got a lot of these, largely isolated by Bill Robbins, and all of them contain a substitution in the tail knob protein GP12. This is telling us that it's the tail that is conferring the specificity for infectivity in the absence of tail fibers. So in the way that these mutants arise, they are frequently multiples, and that's probably simply a result of continued selection for growth under selected conditions. If we look at those mutants, what we find is there are many of them are in 12, some of them infect the adapter protein GP11, a few in this tail protein 7.3, and a couple of oddball guys that we don't understand. It's possible that some of these are here for the ride, have got nothing to do with the phenotype. But one of the things about this phage and its suppressors is if we look at its specific infectivity, how many plaques does this phage give per particle? And here's our value for T7. It's about 5 times 10 to the 11th PFU per OD. Here's one particular fibrous mutant, and it's down by 2 logs. But this is not anything to do with a drop in infectivity per se. Only 1% of the phage particles are making a plaque, but plaques are an artifact. Phages never make plaques except in your laboratory. It's true. And it turns out that it's the viscosity or something, maybe the osmolarity of the soft agar, that is preventing these fibrous phages from making a plaque. And I can show you that in a slightly different way. So here's, phage titers sometimes lie. This is another T7 wannabe phage. It's a salmonella phage called SP6 that I switched its host range, or it's expanded its host range such that it would grow on E. coli. And so here are some salmonella strains, the parent strain to this phage. Here's our E. coli strain. And if we purify this guy, grow it up on E. coli, and we know what the mutations are, plate it out on E. coli, get unit number of plaques, and now we titer that same stock at the same time on different salmonella strains. What we find is that somewhere between 100 and 1,000 particles are making additional plaques relative to the one here. This is the same problem. This guy cannot make plaques in soft agar for whatever reason we don't yet fully understand. Okay, so what are tail fibers good for then? That turns out to be boring. It's the same as in T5 or in lambda, that it increases the rate of adsorption. So here's an experiment. Here's T7 wild type, 95% adsorption within about 30 seconds. You can calculate the adsorption rate constant. And here, look at this time scale. It's in seconds, so it's exaggerated perhaps. But 15,000 seconds is an awful long time for a phage that has this was probably done at 37 degrees, and the lysis time, the latent period of this phage, is less than 20 minutes, which is only 1,200 seconds. So over 10 times as long, and we're still only adsorbing 40% of our input phage in there. So tail fibers are great in terms of speeding up adsorption, but they're not conferring specificity. And we can show that in a different way And here is a K12 cell, and we can use the KO set of mutants to truncate the LPS down to the KDO sugars. And what we can find is that T7 wild type plates on these guys with a largely complete, certainly a complete inner core of LPS. They can tolerate some defects in the outer core. And then you have to get mutants in T7 wild type to grow on the truncated LPSs. Turns out that the same mutations that acquired by wild T7 have to be acquired, or the same overlapping spectrum of mutations are required to allow the fibrillous phage that already has one mutation affecting the tail knob gene to grow on these. So the specificity of a fibrillous phage is being, or of the phage is actually being controlled by the tail and not by the side tail fibers. Okay, so we have a whole bunch of these mutants or substitutions affecting GP12. And since Jun and Zhenyang Wang have got the structure of the T7 tail at atomic resolution, we can map all of the mutations, the substitutions we've got, onto the structure of the tail and say, what are they doing? How do they do their thing? And there's a collection of substitutions that are right at the very tip of the tail itself. These are probably the best suppressors that we have, and these are probably directly interacting with a component of the LPS, which we don't formally know as yet, but is probably right at the very base of the LPS. Oops, I'm going the wrong way. And that is the KDO sugars right next to lipid A. So the tip of the phage tail has to penetrate down through the carbohydrate chains of the LPS to reach its real receptor just outside the lipid A region. So we've got other mutations there coded here. So here's mutations A and here. B mutations map into this region of the structure, C and D and such. Basically, some of the mutations, we're almost done? 2 minutes, okay. Some of the mutations are on the outside of the tail, and they likely affect the absorption of the tail to the lipopolysaccharide as the tail goes down towards the KDO region. And others are in the lumen, and I've run out of time, so I can't tell you much more about those. What we think those are doing are affecting the signal transduction pathway between the tip of the tail when it interacts with KDO and the head, which would be up here, to tell the internal core proteins it's time to go. And I'm going to skip through one more thing here. We've done the same sort of thing with mutants that lacking GP7.3. Again, it's the same story that we've got mutations, substitutions that affect the inside of the lumen of the tail. This is the lumen where DNA and proteins will come through during the infection process. These are probably all reducing the activation energy for infection. And again, some are on the outside of the particle, and I won't say anything more about them at the moment. Let me go back to one more picture here and say the mutants in 7.3 that we isolated have allowed some really rather super work to be done because they also have a defect in infectivity. But rather than just not absorbing, they try to absorb and then they abortively infect. And as a consequence, they the abortive infection leads to dissociation of the phage, leading to released particles that we can then isolate by single particle EM. And if we take now a cryo-ET density map of wild-type T7 infecting cells, we can now dock a structure in there directly, saying that what we're looking at with our mutants actually also reflects reality with wild-type phage. And so I'll leave you with that. All the structural work was done by Chun Yang in June's lab, and the genetics and physiology have taken many years, the major protagonists being Priscilla and Bill and myself when I interfere adequately in the lab. Thank you.
Thank you, Ian, for the great talk, and we have time for questions. Paul.
Have you worked out any kind of an experiment with some of these mutants, Ian, where you can compare EOP to, say, absorption and infection efficiency in solution to see a difference? Because that would help you discriminate between
assays are obviously
failure
done
and absorption
in solution. Absorption assays are always done in solution by definition.
Right. So is it a failure of ejection, or I'm just wondering where the separation between the inadequacy of EOP is and what is really happening in solution?
It's diffusion through the agar to find the right site on the cell surface. I mean, they are hampered. They don't have tail fibers to help them walk. We don't know the details. We have ejection competent or perhaps trigger happy mutants that will infect at low temperature, which is perhaps where you would expect them. We haven't got the same sort of library of mutants that they got in T4, where they've got trigger happies in a different sense there too.
So maybe this is related to the previous discussion, but I'm a little bit confused about what is the energy, the activation energy of infection. Is it one number, and have you done a series of temperatures?
So we measured the activation energy for wild-type T7 infection in vivo, and it's about 28 kilocalories per mole. We have not. It's a huge amount of effort, by the way, to do that. And we have not measured it for any of the mutants. I'm assuming that they have to be lower.
I also have a question. I wonder, when you have mutants that approach the RAF or the core LPS, when I look at all these exchanges, it's rather that you lose negative charges. And as you have magnesium, or bivalent cations that are bound to the charges on the lipopolysaccharide, that could play a role. Could you destabilize your mutants also by pulling off bivalent cations? Are they less stable in EDTA, for example?
Okay, let me back up a second and say that we have not looked at the effects of magnesium or calcium or any of these guys on the absorption process. And I agree, it could really affect. In the answer to your terminal question, these phages are rock stable. You can put them into EDTA and they're happy for years.
No, I was asking because you had more what you call
even the mutants.
Yeah, but you had more abortive infection and particles that had lost DNA.
Yeah, I overran. So those mutants, they bind to cells. They absorb, as far as we can tell, they absorb normally. But many of them, instead of being in a vertical direction, they're actually at an angle. And so when they try to eject proteins and DNA into a cell, it's coming down at an angle to the cell surface. And instead of going into the cell, the phage bounces off. And that's what we see. And we see empty particles Perhaps it was the more interesting part of what I should show, but never mind. It's too late.
Thank you, Ian. Are there more questions? Yeah, and I think we will be happily continue discussions on
Sure.
T7 adsorption and infection and come to our next talk. So our next speaker is, sorry, I just skipped the page here. Sorry, Daniel Hurdiss, and he will speak about, still I'm completely lost on my program. So we start all over again because you married a proper introduction. So Daniel Hurdiss will speak on sweet secrets of common cold coronaviruses.
Okay, hello everyone. My name is Daniel Hurdiss. I'm an assistant professor at Utrecht University. I want to begin by thanking the organisers for giving me the opportunity to present some of our research today, which focuses on the cell entry mechanisms of coronaviruses. So I promise this isn't a SARS-CoV-2 talk. However, I am going to start with an introduction which uses SARS-CoV-2. So for those of you who aren't really that familiar with the architecture of coronavirus spike proteins, these are homotrimeric class 1 fusion proteins, which have an architecture as shown up here in the top left. These can be broadly divided into two parts. We have the S1 part on the left and the S2 part on the right. So this S2 part, shown in red, comprises the actual fusion machinery of the spike protein, and the S1 part can be subdivided into additional domains. In blue, you can see the NTD, or S1A, as we call it in Utrecht, and the green domain, otherwise known as the receptor binding domain, which we call S1B. So when this S1B domain, in green, opens up, it can engage with the host cell receptor, which in the case of SARS-CoV-2 is ACE2. Then, in conjunction with proteolytic cleavage by these host cell proteases, the S1 part of the spike can be removed, and this basically exposes the S2 fusion machinery. This cleavage also liberates the fusion peptide, which can then extend into the membrane of the host cell. And then following some rather dramatic conformational changes in the S2 part of the spike, you can drive together the viral and the host membranes to initiate infection of that host cell. So, given the functional indispensability of the spike protein, it's not surprising that it's the primary target for neutralizing antibodies, and these may be raised in response to natural infection, vaccination, or administered as monoclonal antibody therapies. And actually, the most potent antibodies target this receptor binding domain when it's in this open conformation, and they block it from interacting with the ACE2 receptor. This makes sense. It blocks the receptor, no infection. And we know from tomography studies of the SARS-CoV-2 virion that this virus can spontaneously sample the open conformation of S1B, which in this slide is colored in pink. And this is also corroborated by what I'm sure now is many hundreds of single particle structures of SARS-CoV-2 spike active domains, which you also see this kind of equilibrium between fully closed and open spikes. And actually, this property to spontaneously sample the open conformation of S1B is shared by the closely related SARS coronavirus, which caused the 2002-2003 outbreak and also binds to ACE2. But it's also shared by MERS coronavirus, which binds to a different proteinaceous receptor, DPP4, and periodically jumps from dromedary camels to humans to cause very severe pneumonia. So given the collective notoriety of these three viruses, you would be forgiven for assuming that all coronaviruses can spontaneously adopt the open conformation. But this is actually not the case. So we actually looked at all of the novel coronavirus spike proteins that have been solved across the four recognized genera. And actually, the vast majority of them have only ever been visualized in the fully closed conformation. So this provides somewhat of a conundrum, because on the one hand, the virus has to be able to open to engage its proteinaceous receptor, but it also needs to be able to shield itself from humoral immunity. So ever since these high-resolution structures of spikes started appearing from around 2016, it's been a bit of a puzzle as to how exactly these spike proteins actually open up. So two such viruses that have only been visualized in this fully closed conformation belong to the two human beta coronaviruses which are endemic, and these are OC43 and HKU1. So these are related but entered the human population on two separate occasions. It's thought that OC43 jumped to humans around between 80 and 140 years ago, and more likely for HKU1, this probably happened several centuries ago. And these viruses are typically associated with benign common colds. But if you're very young, very old, or immunocompromised, these can cause quite severe disease. Like SARS-CoV-2, these viruses are believed to have a proteinaceous receptor which binds to the S1B domain. However, the identity of this is not actually known. But this is confusing because how can it engage this proteinaceous receptor if it's fully closed? What is known for OC43 and HKU1 is that they actually bind to 9-O-acetylated sialic acids as a receptor via the S1A domain. So you can see here in orange binding to this S1A domain. And I want to emphasize that in the case of OC43 and HKU1, sialic acid is not merely some kind of attachment factor. It is a genuine receptor, so much so that these viruses encode an additional cell surface glycoprotein called hemagglutinin esterase, which actually cleaves these 9-O-acetyl groups from these receptors. And you can think of these as kind of a two-component system to allow dynamic attachment to sialoglycan receptors, almost analogous to what you see with influenza A and B viruses, which have the hemagglutinin and the receptor-destroying neuraminidase. So the molecular details of the 9-O-acetylated sialic acid binding site have been visualized by cryo-EM for OC43. So you can see here the structure of the spike is closed, and you see that there are two pockets named P1 and P2 into which the 9-O-acetyl and the 5N-acetyl groups insert. However, in the context of a natural receptor present on a cell, this 9-O-acetylated sialic acid isn't going to be on its own in isolation. It's going to be attached to a more complex sugar in the form of a 2,3, a 2,6, or a 2,8 linked sialic acid. But actually, it's been very difficult to define what the actual specificity of these coronaviruses is, because it's very difficult to synthesize these kind of more complex sugars with specific patterns of acetylation. But quite recently, my colleagues in Utrecht came up with a fairly ingenious chemoenzymatic synthesis pathway in which they actually used the virus's own enzymes to synthesize these receptors. And by doing this, they were able to generate a whole range of different sugars with specific acetylation patterns. And using this, they could generate a glycan microarray. And this demonstrated that OC43, compared to its presumptive ancestor, bovine coronavirus, is starting to develop a preference for this 9-O-acetylated alpha-2,8 linked sialic acid. And HKU1, which has been in the population for much longer, has developed what you could say is almost an exclusivity for this receptor. And I think this is a really nice example of kind of convergent evolution that we're seeing. So at the start of this project, we had a very straightforward question. What is the structural basis for this alpha-2,8 linked sialic acid preference? Straightforward question. Before I jump into the results, I just need to mention that there are actually 3 recognized genotypes of HKU1, but for all intents and purposes, there are 2 distinct serotypes of the spike, and these share around 84% sequence identity. So the structure of the HKU1B spike was resolved by the groups of Andrew Ward and Jason McClellan in 2016. This was the first human coronavirus spike protein to be characterized long before SARS-CoV-2. But the structure of the HKU1A spike, which is what we actually used in our glycan microarray experiments, had not yet been determined. So we thought, before adding the sugar, we first wanted to determine an APO HKU1A spike structure. So we went ahead and did this, and we determined the structure of HKU1A spike at around 3.4 angstrom resolution, and this allowed us to really nicely model all of the different domains which make up the architecture of this protein. And quite nicely, you see the parts here in yellow. These are actually parts which were missing in the HKU1B structure because they were quite fuzzy or not well resolved. So this is now the most complete HKU1 spike structure that is available. Great. So how does this sialic acid binding site look between HKU1A and HKU1B? So this result was actually a little bit surprising to us. So we know, obviously, from the genetic information that the residues involved, which are critical for 9-O-acetylated sialic acid binding, so you have this tryptophan 89, which basically separates these two pockets for the 9-O and 5N acetyl groups. We have a lysine, which binds to the carboxylate, and we have this threonine or a serine in HKU1A and B, which also binds to this terminal sialic acid. But actually, the HKU1A has this much more shallow bowl-shaped topology, which is a bit weird for a sialic acid binding site, whereas in HKU1B, this looks more like what you would expect for an actual pocket. So we thought this was a little bit weird, but we know that our HKU1A spike binds to the sugar from our glycan microarray experiments, so we just went ahead and did the structures. So on the left, you can see our APO spike, and what we kind of expected was that we would get, like OC43, a closed conformation of the spike bound to the sugar. But to our surprise, upon incubation with our alpha-2-8-linked 9-O-acetylated sialic acid, we saw a surprising heterogeneity in the conformations that we observed. Around 25% of the particles remained closed, and you can see here in pink, this is the sialic acid. Around 20% had converted into this one-up conformation, and the majority of the particles were now in this fully open conformation, which led us to hypothesize that adding this sugar to the S1A domain is allosterically triggering opening of the S1B domain. So to kind of exclude the possibility that this was some kind of strange pH effect from mixing the sugar with the spike protein, we actually made a mutation in the sialic acid binding site which completely knocks out binding of this sialic acid receptor. We redid the cryo-EM experiments with the sugar, and then you see no opening of the spikes. The population is, what we can image, is 100% closed. So what I'm showing you here is a morph between the apo structure, which is shown here in yellow, and this will morph to the hollow structure. So one thing that you can notice very nicely in this circled region is the appearance of this nice peanut-shaped density, which is our alpha-2-8-linked sialic acid. So these are the two rings. But we also saw a very conspicuous conformational change in this E1 loop, which flanks the sialic acid binding site. And it also seems that there is some kind of ordering of this N29 glycan in this loop, which seems to be concomitant with this conformational change. So this site is inherently quite flexible, but the resolution was just good enough for us to actually have a go at modeling this. So you can see here on the left, we have our apo structure. And then you can see here on the right, somewhat restrained by what we know about the allowed stereochemistry of these glycans and what we know about these sialic acid binding sites, we modeled our alpha-2-8-linked glycan in here. So you have the terminal sialic acid with the 5N and the 9O pointing into the P1 and the P2 pockets, and we orientated this penultimate sialic acid so that it's 5N acetyl groups facing towards this E2 loop. And indeed, you can see that there is this ordering of this N29 glycan and this shift of this E1 loop, which is now partially covering this W89 residue, which separates these two pockets. So what does this look like when we morph between these two structures? So you can see that this E1 loop, colored here in yellow, actually has quite a large conformational change which propagates down this strand here. And this actually brings this threonine 30 in proximity to this tryptophan 89, which actually stabilizes the orientation of this tryptophan and which actually stabilizes this sialic acid binding pocket. Now, because of the somewhat limited resolution in our cryo-EM reconstructions, we wanted to corroborate our findings with an additional technique. So for this, we turned to all-atom molecular dynamics simulations. So our experimental setup was as follows. We started with the apo structure of this S1A domain. We then place our alpha-2-8-linked sialic acid into the binding pocket, and we restrain this based on what we know about how this terminal sialic acid can bind into these P1 and P2 pockets. And then we did a whole lot of simulations. So for people who are familiar with MD, we did about 70 microseconds across all of the experiments, which is quite a lot. And I won't play the entire video because I think I will definitely go over my time if I do. But the point I want to show is that upon simulation of this system, you start to see these conformational changes, and what you might have just noticed is actually this conformational change propagates down this strand, and you actually see this register shift between these two strands. So Zooming ahead, so this is what this simulation actually looks like when it finishes. So this is after 500 nanosecond simulation, and you can see that what we visualize here is actually it faithfully recapitulates our cryo-EM results, which I think is a really powerful example of how you can combine these two techniques to support one another. So you can see, fully consistent with our EM data, we have this shift in this E1 loop. You have threonine 30, which can now hydrogen bond with W89. Serine 86 faces its side chain out of this P1 pocket to interact with this backbone of leucine 28. The proline 33 completely shifts its orientation, and this basically helps to stabilize the P2 pocket. And in full accordance with what we see in the cryo-EM, we see this register shift so that actually D76 and F47 actually exchange their hydrogen bonding partners. So this entire conformational change requires the breaking of two hydrogen bonds and their reformation with new partners. So what does this mean in terms of this whole allosteric mechanism? So if we zoom out a little bit so that we can see the entire S1A domain, if you keep your eyes focused on the yellow strand, which is E1, and the adjacent black strand, you can see that this register shift actually drives an inwards rotation of the entire upper half of the S1A domain. So you have this intra-domain wedging, which seems to be driven by sialoglycan binding, which is pretty cool. And if we zoom out a little bit further in terms of how this could actually trigger opening of this receptor binding domain, what happens is this inwards movement of the S1A, which is shown here in gray, actually completely reshapes the interface between the adjacent closed S1B domain shown in orange, and we believe that this weakens the interface sufficiently so that it can adopt this open conformation. So putting all of this information together, we were able to propose a stepwise series of events. So we have sialoglycan, which binds to this S1A pocket, which leads to conformational changes in the S1A domain, which in turn lead to opening of one and then subsequently the remaining S1B domains and opens the spike up. So this allows us to propose the following model, whereby our HKU1 spike approaches a host cell membrane. We know that coronavirus spike proteins can tilt quite significantly from tomography studies. It binds to a primary receptor, which is probably a 9-O-acetylated ganglioside like GD3. This binds to S1A, allosterically triggers opening of S1B, and based on what we know in the literature, this probably is binding to a secondary receptor. So first and foremost, this data fills in a critical part of the HKU1 cell entry puzzles. But taking a step back, so from a more general perspective, these results actually show that coronavirus entry might be much more complicated than what we actually currently believe, with possibilities of co-receptor engagement. So for example, with HIV, which binds to both CD4 and CCR5. So you have this kind of elaborate entry mechanisms. And I think this will kind of really spur a lot more studies for people who will start to look for what we now term these primary receptors which trigger opening of the S1B domain. So that brings me to the end of my talk. I'd like to thank all of the people involved in this research, in particular my co-senior author, Raoul de Groot, who's actually been working on coronaviruses since before I was born. So I've learned quite a lot from him after collaborating for a few years. First author Matti, who worked really, really hard on this project. Our collaborator Martin Frank, who really is an MD wizard, and he really did a great job with the MD simulations. And also Jeva, my favorite collaborator, who gave us access and provided data collection for the HKU1 cryo-EM, as well as all the organizations which provided funding or technical support to make this work possible. And I'd like to thank all of you for your attention, and I'd be happy to answer any questions.
Thank you, Daniel, for the really beautiful example where we can see that cryo techniques can now also address ligand binding situation, especially with glycans, which is very important. I think there are a lot of questions.
Maybe that was a link to the phage stuff, so yeah, tenuous.
I had what if you were to mix sialic acid in substoichiometric amounts? Do you then think that the RBDs would open up sort of cooperatively, like the opening of one is enough to get the others to open, even without because physiologically you can't have, I wonder, whether you'd have all three sialic acids.
No, this is a really good point because, yes, in the context of, there's no possible way that the spike can orientate all three S1A domains so that it can simultaneously engage all of them. So it would be interesting to try and do maybe sub-stoichiometric amounts of the sugar, or maybe, if you wanted to go a little bit crazy, actually do tomography studies binding to cells which are decorated with these nanoacetylated sugars. But we did do an additional time point, which I didn't show in the talk, where we basically did a 1 minute incubation, and then you only had one S1B opening. So we can now kind of really piece together that, over time, more are opening, and this is probably not reversible like you see in S1B for SARS-CoV. Yeah.
I was wondering whether the additional N-glycan would play a role. I mean, you need the energy from the binding to get this register change.
Yes. Yeah.
And probably would also stabilize the final complex and you showed that you'd suddenly see that the N-glycan becomes ordered.
Yeah.
So what would happen if the N-glycan is missing, or when there are heterogeneities in this glycosylation patterns which usually occur in these spikes?
Yeah, so there are previous studies from my colleagues where they did kind of mutagenesis experiments, and they showed if you knock out this N29 glycan, this kind of like abrogates the entry. So, but they couldn't really put that into any context. But now we know that, yeah, it does seem that this glycan in this final stable conformation that it adopts really probably seems to be stabilizing this alternative topology of the sialic acid binding site. It's probably also worth mentioning that we did a lot of control MD simulations, so we did them also in the absence of the sugar to see what happens. And actually then these kind of shifting conformational changes, they can occur, but very sporadically, and they're not stable. So it will kind of transiently sample the shifted topology and then kind of spring back. But then if you put the sugar in there, it really, after about 500 nanoseconds, it becomes a single stable conformation, which I think is pretty nice. Yeah.
Sorry, another crazy question, Dan.
First of all, over there. Yeah.
Hi, here.
Hey.
I know that the N-terminal part is much less variable than the RBD, but have you checked for the mutations in the binding site that could alter the specificity or something like that? Because the sialic acid cannot change.
Yeah, so
much,
it has to keep these kind of core sialic acid binding residues. So again, I couldn't show this, but in a separate study, which was running parallel to this, we were also doing antibody discovery against OC43, which is the other nanoacetylated sialic acid using Embecco-virus. And in that study, what we actually showed is that antibodies which target cryptic parts of the S1B domain, if you challenge these viruses with these antibodies, they gain resistance mutations in this E1 loop. So it seems that they're somehow obtaining mutations which can allow them to kind of modulate where and when they expose this S1B domain. So we thought that was a really nice independent experiment which kind of shows that there are mutations which are not in the site but close to it, which can kind of modulate this opening and closing. So it kind of makes us feel that this is a general mechanism for these coronaviruses.
I think there was one more question in the back.
It was just a crazy question, but if you were to put the sialic acid binding site on SARS-CoV-2 spike, do you think it would then stop opening?
So actually, this is a good point. SARS-CoV-2 does bind sialic acid. There are studies which show that it can bind sialic acid, but the relevance of that is not so firmly established for these viruses. So our hypothesis is that many of these coronaviruses that we showed in their natural host populations, they've co-evolved in the presence of humoral immunity, and therefore the emphasis is really on staying closed. So they've probably evolved specific triggers which allow them to pop open their S1B domain so that they can bind to the receptor. When a virus enters an immunologically naive population, such as in the case of SARS, SARS-CoV-2, or MERS, it's open season on receptor binding because there are no antibodies to worry about, right? So, and it's actually known that one of the first mutations that SARS-CoV-2 obtained was the D614G mutation, which actually pushes it more towards the open conformation. But now, if you look at Omicron, it's starting to move more towards a closed conformation. So we think that this spontaneous opening is an evolvable trait. So in the future, maybe SARS-CoV-2, as it kind of completes its transition to endemicity, will become fully closed again, but then it would have to kind of engineer a primary receptor. Yeah.
Yeah, thanks again, Daniel. And we're now moving back to phage. So we came from phage, we moved to virus, we go back to phage. And Taylor Lindner will now speak about a Shigella virus. So it's a bacteriophage and a critical carbohydrate for attachment.
Okay. Hi, I'm Taylor. I'm a PhD student in Sarah Doerr's lab at the University of Florida, and I am just finishing up my second year. So please bear with me as we go through this very difficult material. So Shigella flexneri is a gram-negative rod-shaped enteric bacteria. It can cause shigellosis, which, as we know, is a form of bacillary dysentery. Has symptoms including severe diarrhea, fever, stomach pain, nausea, and vomiting. Definitely not a fun time. The specific strain that I'm working with is CFS 100. It is Shigella flexneri serotype 2A, and it's a 2457T derivative. So first off, why do we care about Shigella whenever we have all of these other bacteria? So first off, Shigella flexneri is the leading cause of shigellosis out of all of the Shigella species, and it causes roughly 188 million cases and 164,000 deaths annually. Antibiotic resistance is on the rise. We know it, we hear it all the time, but what can we do about it, right? And interestingly enough, the Shiga toxin genes can be transferred horizontally to non-pathogenic Shigella cells as well as E. coli. And phages can be a vector for the transfer of these genes as well as antibiotic resistance genes. So E. coli and Shigella are extremely similar bacteria, and there are many characterized phages that use E. coli as their primary target, but there are a few that have been characterized that use Shigella. And with the similarities between these two bacteria, you would expect that there would be at least a large amount of overlap between these phage, with an E. coli phage being able to target and infect Shigella and vice versa. However, that's not the case. There are many significant differences between both of these phage types, and that results in very few phages being able to cross-infect the other bacteria. So SF14 is a double-stranded DNA myovirus with a T=9 capsid geometry. It's relatively new. It was first isolated from Red Cedar River in East Lansing, Michigan in 2016 by the parent lab at Michigan State. And the work that has been done so far has shown that it will infect multiple serotypes of Shigella flexneri, but not multiple Shigella species. So when most gram-negative bacteriophage begin their infection cycle, they will look for a primary receptor that they will reversibly bind to. And then once they find that, they will then go through the process of looking for a secondary receptor. If they find it, they will then irreversibly bind and go through its process of ejecting its genome. So serotypes are determined by LPS composition and pattern. And this is, as you can see, an LPS molecule. The lipid A is what helps anchor it into the outer membrane of the cells. And both the inner core and the lipid A are conserved between bacterial species. The outer core is mostly conserved within the species, and then the O antigen is highly variable. And when speaking about Shigella bacteria, it's primarily only found in Shigella flexneri. So, as I mentioned before, with the primary receptor, this is often a carbohydrate found in the LPS. And so to begin narrowing our search, instead of looking at every single thing, we decided to focus primarily on the outer core genes. We made spot tests after we used. I apologize. I am very, very sleepy and very nervous. Thank you. But we made mutant strains of these genes. So we made a delta WAL, WAK, WAJ, WAO, WAG, and WAC, a lot of WAs. And so we performed spot tests with all of these, and the only delta strain to lice was WAL. And so with that in mind, we decided to focus on the furthermost outer core genes, so L, K, and J. We decided to leave O alone because it kind of grows sad. It's very small. And interestingly enough, the lyse regions on the delta Wa-L plates were larger than the ones found on the parent CFS 100 plates. And you can see it here, but Wa-L is responsible for the oetogen being added onto LPS, Wa-K is the N-acetyl glucosamine, and Wa-J, the glucose 2. So our current hypothesis is that SF14 is using one of the outer core carbohydrates and Shigella flexneri as the primary receptor due to the pattern of LPS conservation. So we used homologous recombination to replace our genes of interest that I had mentioned before with a kanamycin-resistant cassette. And then this gene segment was confirmed with PCR and gel electrophoresis both before and after transformation. And then we sent all of these strains off for further confirmation that our kanamycin cassette is where we wanted it to be. So because we were looking for a primary receptor, we decided to perform attachment assays and potassium efflux assays to track initial binding and genome ejection, respectively, within the SF14 infection cycle. And yes, I do remember Bentley yesterday saying, why? He doesn't do efflux assays, and it's fair. Not anymore, yeah, it's fair. We had to go through, we had to buy a whole new probe because we thought ours was damaged, so we began with the attachment assays first, and just like a quick run through of how we did it, we inoculated 10 milliliters of LB broth with 300 microliters of overnights and added kanamycin to the ones that have the knockouts. And then we inoculated with an MOI of 0.1 to prevent the cells from becoming overwhelmed, but also making sure we had enough phage that we could reliably detect attachment. And then we took samples every 5 minutes from 0 to 20 minutes, and then those samples were immediately spun down in a centrifuge to prevent any unattached phage from attaching. And then as a secondary precautionary step, as soon as those samples came out of the centrifuge, we immediately diluted them as well. So I just put this over here just as like a little reminder. But as you can see with the CFS 100, very nice attachment, almost complete attachment by 15 minutes. The Delta Wa-K strain attached very similarly, which kind of expected at least a little bit, considering that it did not lyse. Same with Wa-L. And if you look closely, you can see that it had a faster attachment than CFS 100, which again, is kind of understandable. It had a larger evidence of lysis, like larger plaques, everything like that. However, SF14 does not like Wa-J. It's constantly detaching. It doesn't want to stay bound. So that was kind of a big, look at me kind of sign. So with that, we have so far determined that SF14 attaches to delta Wa-K and delta Wa-L similarly, if not to a higher standard than the wild-type CFS 100. SF14 attaches very poorly to delta Wa-J and detaches much more readily. And so we have so far determined that glucose 2 appears to be important as a primary receptor for SF14. We still have more tests to further prove this, however. So with potassium efflux assay, we are measuring potassium ions as a proxy for genome ejection due to the cell membrane permeability. So as the phage genome enters the cell, the damage done to the membrane, as well as the cell trying to maintain its homeostatic and isotonic balance with its environment, will release potassium ions. So we grew our cultures to an OD of 0.5, and then we inoculated them after a series of centrifugations and resuspensions with an MOI of 5. So the very first time point 0, was taken before phage was added, and then this next time point was taken immediately after adding the phage. As you can see, there's like that initial increase of potassium ions from when the phage was added, and then you see a steady increase in potassium ion release. Wa-K, once the phage has been added, you have that little jump, and then it stays relatively steady, doesn't increase very much. While L, we're unsure of why it had such a lower initial reading, but very, very large jump, much faster than CFS 100, and the final values are around the same for both of those strains. We think that the reason it had such a large jump was because the O antigen is missing, so it's allowing the phage has an easier attempt to locate and bind to both the primary receptor and the secondary receptor. And like expected, once WAJ had the phage added to it, stays relatively steady, no genome ejection really going on. So with that, although CFS 100, delta YL, they had the same relative end release of potassium, but YL had a much faster release. The potassium ions released from delta YK and delta YJ maintain those steady values as predicted based on the lack of lysis. And again, we're unsure why delta YL and delta YJ had those much lower initial readings. So as a general wrap up with the data, glucose 2 appears to be important for SF14 attachment. Removing Shigella flexonary's O antigen appears to increase SF14 attachment speed. And the N-acetylglucosamine seems to have a part in the infection process as well, based on the fact that it's not lysing, but it is attaching very well. Oops, sorry. So some of our near future and already beginning experiments, we are planning to perform both the attachment and the efflux assays on the delta strains YO, YG, and YC, just to kind of help show, hey, the primary receptor is further up in the LPS. It's not closer to the lipid A. And then we're also going to create more of the LPS biosynthesis gene knockout strains not currently in the lab, such as delta YD and delta YB. And then we are currently beginning preliminary attempts to identify the secondary receptor for SF14 using mini cells. Some more distant future experiments that I'm planning, I want to create double knockout strains and test those as well, specifically a YL-YJ double knockout and a YL-YK double knockout. And then I want to also perform more experiments with complementary strains of both the single knockouts and the double knockouts. So I want to say thank you to my ever-patient PI, Sarah, all of my lab members, the Flippies pals who make sure to drag me out of lab so I don't drown in paperwork and papers, but instead might drown under the water. And I'd like to thank my committee members, and thank you for listening.
Yeah, thank you for the nice presentation.
Thank you.
I think there's a lot of work to do, but you have still some years to go in your PhD. Other questions?
Is there much known about what the LPS looks like in E. coli and the non-susceptible Shigella strains? Could you get any clues by comparing the LPS patterns between susceptible and non-susceptible species?
Susceptible, non-susceptible, like Shigella species, and then SF4, like Shigella's flexneri, because I heard you mention E. coli. Did you mean to say that one as well?
It's also not able to infect E. coli, right? Yeah, so just anything that's susceptible versus anything that's not susceptible.
Yeah, so I don't know the full details off the top of my head, but let me go all the way back. So the main differences between the Shigella species themselves is going to be in this outer core, like the specific order and composition of it. Definitely the O antigen with the Shigella species, other than Shigella flexneri, Sonii will occasionally have an O antigen present, but the main differences between those are going to be in the outer core. And I'm not sure about the differences between E. coli and Shigella flexneri LPS. However, I would definitely say that the outer core is going to be different, and the inner core and lipid A will likely be different, but I don't think they'll be totally different. They'll probably be like crossovers and similarities between both of those, though.
How did you measure the potassium efflux?
Sorry?
How did you measure it? With radioactivity or?
We used an isotonic low-level ISA to kind of like get a background reading on the buffer we were using, and just use a potassium probe and just kind of measured like the change in millivoltage and stuff like that, and then kind of converted from there. I'm not really sure how you want me to explain that. I'm sorry.
Are there any more questions? So let's thank Taylor again for the talk. And again, we switch from phage to virus, and now the virus is hepatitis B. And the talk will be by Jodi Hadden-Perilla on new conformations of the hepatitis B virus capsid protein revealed by all-atom molecular dynamic simulations.
Hi everyone, my name is Jodi Hadden-Perilla, and I'm an assistant professor in the Department of Chemistry and Biochemistry at the University of Delaware. And yes, I'm switching back to virus, and I'm happy to be here today to tell you about some of my group's recent work on the hepatitis B virus capsid using all-atom molecular dynamic simulations of the intact capsid to reveal new conformations of the capsid protein, or what we're really doing is sort of revealing a more comprehensive ensemble of conformers that are possible in the equilibrium dynamics of the capsid. So just a quick anatomy of hepatitis B. It's an enveloped virus. In its mature form, has a DNA-containing icosahedral capsid. It's a T=4 icosahedron. In its immature form, it contains pregenomic RNA. If we see down at the bottom here, if I can figure out how to work the laser, this is a T=4, so there's 240 constituent proteins. There are 120 dimers is what we really have. We call them the A, B, and C, D dimers. The A chains form capsid pentamers, and the B, C, and D chains form capsid hexamers. Just a quick look at the viral life cycle will show you just how often the capsid participates in the replication cycle. Has a lot of very key functional roles that it plays. It's a very multifunctional protein, and for that reason it's of great interest as a therapeutic target. So ever since I was a postdoc, I've been running simulations of the intact hepatitis B virus capsid. So this is based on a crystallographic structure that's been available since the 1990s. But these crystal structures, we get this idea because they're symmetrized, that they're sort of this perfectly symmetric shape, and maybe they're sort of rigid. And with these types of simulations where we remove symmetry constraints, we put the capsid in water, we just sort of let it relax and move and wiggle and jiggle. We just release it into the wild and kind of finally get to observe what it's going to do, and we find that it's actually remarkably flexible. In fact, this is an incredibly flexible capsid. This is just a note to say that we have this capsid in a representation of native conditions. It's in an explicit solvent box with pH 7 and a physiological concentration of salt, and altogether this represents 6 million particles. This original simulation took about 7 months to equilibrate and sample 1 microsecond on what was at the time the largest supercomputer on any university campus in the world. Very expensive, but I promise you I need all of the atoms. I'm going to show you some of the reasons why. All right, so this is some of the reasons why. So this is a variety of studies we've done over the years using our trajectories of the intact capsids, both the apoform capsid and those complexed with capsid assembly modulators. We've learned a lot about the morphology of the capsid, how incredibly flexible it is, mutations that affect assembly or confer drug resistance, symmetry breaking of the capsid. We've helped some of our experimental collaborators interpret some of the data that they've collected. We've been able to track the movement of individual ions in water and look at the exchange rates of the capsid, how drugs affect its morphology. I think we have under review right now our 8th paper that's utilized data from this trajectory, and we'll have one more as soon as I get home to edit the first manuscript. I've let a graduate student write completely by themselves, so we'll see how that goes. Please wish me luck. So today I have 3 stories for you that are related to recent work we've been doing. This first story is on the intradimer interface. So this dimer kind of looks like this. Okay, so a monomer looks like this. There are two of them, and they assemble back to back, and that's the dimer that you see there. And so this intra-dimer interface where the two monomers assemble, that's the part I'm talking about right now. And if you saw on a previous slide, this capsid is sort of a spiky ball. This part that sticks up where my fingers are, this is the spike. And we were running simulations of the apoform capsid as well as the capsid complex with the phenylpropenamide AT130, and we were tracking various conformational properties just to sort of see how they might differ between the two systems. And one of the things we tracked was a hinge angle. So back to this silly hand display here, there's a hinge right there where the spike tip can be quite flexible. It's not bent too much in any of the crystal structures. We were tracking some of these, or tracking the dynamics of this hinge, and in the apoform capsid, we have the B, C, and D distributions of hinge angles looking very similar. Those chains are all in hexamers. The A chains make up the pentamers, so we could attribute this difference to quasi-equivalence. When AT130 is bound, all of these distributions shift. And we started looking at the differences and what was really underlying the structures that are contributing to these distributions. We became really interested in this shoulder here that we were observing in the D chain, where we have a higher probability to have larger hinge angles. And so just for some context, these are what the dimers looks like most of the time. They have this really nice intact intra-dimer interface. So right here in the spike tip, the spike is closed. These helices are well folded and nicely interacting. Occasionally you have some disorder in the spike tips, but the interface is quite preserved. We started to see some really wild conformations when we started digging in this ensemble though. And one of the wildest ones we found were structures like this where the spike is completely peeled open and it's exposing hydrophobic residues. It can even make this salt bridge, which had never been observed in crystal structures before. Normally these residues are quite far apart. Turns out the base of the 4-helix bundle can also pry open, which lifts the fulcrum to make this salt bridge. The whole fulcrum can shift up to form this salt bridge. We observed this open conformation with D or C chains being bent, but we never observed it in an AB dimer. This was more common in our AT130 bound capsid, but was also occurring in the apoform capsid. So we became really interested in this open conformation. What does it mean? These viruses are so highly evolved and very specific to their hosts. They tend not to do things sort of on accident. Probably this has some function, but what is it? One idea is that this is an assembly inactive dimer. There's this integrity of the intradimer interface is known to be important for hepatitis B capsid assembly, so it could be that this open conformation is an assembly inactive state. It's been proposed there's a conformational switch that activates assembly. Maybe this is part of that. We took it out of the capsid, one of these open conformations, and ran it isolated in solution to see if maybe constraints from the capsid lattice were stabilizing this open conformation. But it's quite happy to be open, surprisingly. It stays this way for hundreds of nanoseconds. Even after the salt bridge breaks, it stays open. So we're not really sure if this conformation is relevant to free dimer or in capsid or both, or really even how common it is in a truly converged equilibrium ensemble. Another idea we have about what it does is because this is opening and exposing this hydrophobic patch, maybe it's a host factor binding site. We started looking into what binds the spike. There have been some recent reports that spike can interact with CRM1, chromosome region maintenance protein 1, also known as exportin 1. It's a nuclear export protein that's been shown to perhaps export immature HBV particles or from the nucleus, to export them from the nucleus, or to secrete mature particles. And we started looking at the sequence of our protein, and we found several consensus patterns for nuclear export signals. The two of them with the stars became quite interesting to us. We started to mine this massive conformational ensemble we have for all these different conformational states that we've observed the dimer can adopt. And we've been able to build some models of our capsid bound to CRM1. We align in all of our conformations. I think we've used 12 million different conformations in this study. We relax them, we throw away anything that has a clash, and then we save the rest. And we've observed 2 different binding modes that seem to work. So importantly, you can have negative and positive sense nuclear export signals. You need this helix strand secondary structure. This strand bit, you need it to fit down in this deep P4 pocket. you can have this strand be at N or C terminal. This NES signal is exposed when the spike is partially disordered at the top, so you can get the strand at the spike tip. That does not require this open dimer conformation. However, when the dimer is open and that hinge is dramatically bent and it's disordered and the hinge can actually form a bit of a strand, you can actually get this positive NES signal that's exposed and can also bind according to our modeling. So we're looking more into that. The next story I have for you involves the six-fold pore, which is a pore in the middle of the capsid hexamers. Start a few of them here for reference, as well as the capsid linker domain. So we've been running simulations of the assembly domain of the capsid, also known as CP149. It's missing the arginine-rich domain that would bind the RNA and interact with some of the other host factors, but we retain the linker, which would connect those two domains so that the end of our protein encompasses the linker, which the terminal residue of that is 149. And so what happened was our collaborator Adam Zlotnick has a cryo-EM reconstruction of important beta bound to the capsid, and it's bound by contacting the arginine-rich domain, which is extended through the hexameric pore. And this is what his hexamer looks like. And he was wondering, is this pore dilated? There were questions about the mechanism of how that arginine-rich domain gets exposed through that six-fold pore. Does it require dilation of the pore? And because he had a pore diameter that's larger than some that have been reported in previous structures. So we dug into our simulations. We have the equivalent of 30 microseconds of sampling for the capsid hexamer. If you see in this orange distribution, these are all the diameters we sampled for all the conformers that we have, and so it ranges from 4 to about 12 angstroms. So something around 10 is a little large, but it's well within equilibrium fluctuations of what the capsid normally does. Another thing that we observed is that the terminal of our linker domain, the end of CP149, can actually stick through the pore. We observed this in conformations during the simulation, so we got to really ask the question, well, if a peptide is in the pore, does that cause the pore to respond, or does it dilate? Does the pore care that there's a peptide there? And the answer is no, it doesn't seem to. We have instances where there's a single copy of the linker domain extended through the hexameric pore. We have this blue distribution here. It's still a nice Gaussian distribution ranging from about 4 to 12 angstroms. We also had some conformations where there were 2 linker residues poking through the pore. We have many fewer of those, so the distribution doesn't look as nice. But it's still ranging from that 4 to 12 angstroms. So it appears that the dilation of the pore is not required for exposure, at least not of the linker domain. Of course, we don't have the arginine-rich domain in the simulation here, so we can't be 100% sure. Here's some images of what this, of our minimum pore diameter and maximum pore diameter, just some examples of conformations and how they look when these linker domain terminals are extended or when we have 2 extended. We observed a quasi-equivalent preference that D chains tended to be exposed more often, and D chains were the only ones we observed that could be out 2 at a time, and this was consistent with a trend that the experimentalists observed from their cryo-EM data, so it's nice to see the correlation there. And the last story I have for you today involves the inter-dimer interface. So it's the blue one in the figure, it's where my thumbs are, it's where dimers contact each other in the capsid lattice, so inter-subunit contacts. Within this inter-subunit contact is a well-known drug target for the hepatitis B virus capsid. It binds capsid assembly modulators. And so what we've been doing is looking at the dynamics of that binding site. And here's an example of how we're doing that. We're measuring the volume of the site, of the hydrophobic pocket that's within the interface there. We're doing it with the method we developed a few years ago to measure the internal space of biological containers. We originally developed it to look at the interior space of capsid proteins or viral envelopes or large biological containers. My group has been tuning it to be really applicable to small spaces. It allows you to physically delineate the boundaries of the binding site. And here are some examples from crystal structures available in the literature of some CAM-bound capsids and what the volumes of that quasi-equivalent B and C site look like in general. Now, if we look at the distributions we get from MD simulations, we learn that the crystal structure tells you only a part of the story. So in our apoform capsid, here are the volumes in the crystal structure of our A and B quasi-equivalent sites. But after we allow the capsid to relax, we see that there's actually a larger range of volumes that these two sites can adopt, and they are quasi-equivalent. They are not exactly the same. They have different volume distributions. More interesting is in the HAP1 bound case. So this HAP1 bound crystal is actually crystallized, essentially saturated with HAP1 in B and C sites. But the resolution of the molecule in the B site was too poor to get coordinates, so the simulation actually only has HAP1 in the C site. And you can see here that the volume of that pocket at the interdimer interface that's observed in the crystal is pretty representative of the average that we see in terms of our simulation distribution. That B site relaxes though, because now it's an apo site. It doesn't have HAP in it anymore. And we see that this apo B site is different than this apo B site because this C site is actually filled with a HAP. And so this may partially underlie some of the cooperative effects that have been seen in HAP uptake in capsids observed in experiments. I have a movie here just to show you sort of how these volumes of the binding sites are changing over time. We get a lot of motion, very, very dynamic binding sites. We actually even have, I think, 4 copies of our AT130 fall out of these very mobile binding sites over the course of simulation. This is just to show and just to point out that we have binding sites that range in volume from essentially 0 angstroms or fully closed binding sites to something that has the volume or the space between dimers that's essentially twice as much as been ever observed in experimental structures. So these are new conformations that have not been isolated before that allow us to get a better idea of the full range of what's possible in terms of the conformational space of the capsid protein. And for my last slide here, I'll point out something else we're doing with that volumetric data. So we have all these maps of the volumes of binding sites, the empty space in the binding site. And we've been aligning them, superimposing them, and calculating not an occupancy, but a vacancy, a probability that something is a vacant space. So we can use different probability thresholds to look at these maps. And so if we look at voxels that are vacant or part of the binding site, not occluded, 80% of the time we get these volume maps you see far on the right. Remember, HAP1 is actually in this C site, and it's shaped a lot like that HAP molecule. That makes sense. The HAP is sort of holding that binding site open. This B doesn't actually have this HAP molecule in it during the simulation. We've aligned it in. Remember, when HAP is bound in this site, that distribution of volumes for the empty B site changes. In fact, it looks like it becomes shaped a lot more like the HAP molecule. This might be part of, again, this cooperative effect of HAP binding that the experimentalists see. We think that doing these sort of reconstructions of the equilibrium shapes of the binding site might be very useful for future small molecule drug design. So with that, I'll just acknowledge everybody who contributed to the project. So we have a lot of great experimental collaborators as well as computational collaborators that work with us on method development. Most of the work you've seen here was done by my graduate student Carolina Perez-Segura and Rasanjali Lakshani Ranawaka. We have previously been funded by NIH for our HBV work. We're primarily funded now by National Science Foundation, and of course we have to acknowledge all the supercomputer time that has fueled this research over the course of the past 8 years or so.
Yeah, thank you very much for bringing dynamics in all these super frozen structures of this conference today.
Thank you.
And I think there are a lot of questions.
Yeah, just the conformational changes you're seeing in the dimer, is this pre-reverse transcriptase, RNA or DNA phase? It's complicated in HBV.
So this capsid is empty. Does that answer your question?
Yeah. Okay.
Yeah, it's completely empty. So yeah, I mean, once we get some genome in there, all bets are off. We don't know. We don't know yet. So I like to think that we're sort of just seeing now what possibilities are and getting a sense of conformations we might see once we get genome inside. I actually even had a nice breakfast conversation with one of you a few days ago where we talked about, it might be that there are ways that the capsid signals its maturity by transducing some signal from the inside to the outside. Maybe something like that could, where the genome sort of stabilizes that conformation or causes it to be preferred in a way that we don't know yet. yeah, one day we'll have good structures of genomes that aren't symmetrized, and we can build them into our models. Please get to work on that with all that cryo-EM.
So Jodi, what would happen if you added 5 million atoms more of lipid? What do you think it would happen with the dynamics?
Oh, that's a good question. One day I do want to put this thing in an envelope. I'm not really sure what's going to happen. I like to think that a lot of these dynamics will be preserved. I don't know if there'll be any kind of interaction with the membrane. I haven't even thought about that. I guess we'll have to wait to find out a few more PVAs.
Can you comment on the solvent exchange? It's here. Can you comment on the solvent exchange? And for example, if you see the first hydration shell very static and diffusion, water diffusion.
Yeah, so we're actually doing some work with the solvation shell right now for a separate project. So I can't comment on that because we're not finished with it. In terms of solvent exchange, though, this capsid, because it's very porous, the pores are quite large, has pretty rapid solvent exchange. Let's see, off the top of my head, I want to say it's like 4,500 water per nanosecond going in and out at equilibrium. More interesting is what the ions are doing. Ions, sodium and chloride, are also passing in and out of the pores. Sodium exchanges at a rate 5 times that of chloride. It preferentially translocates sodium. It also causes sodium to sort of localize along the inner shell where the arginine-rich domain should be most of the time. So this is evidence that the capsid is sort of controlling its environment around it or influencing the environment. For comparison, so this capsid preferentially translocates positively charged species, at least that's what our ions suggest. The HIV capsid prefers negatively charged species, so it translocates chloride twice as fast as sodium. So, different capsids, different mechanisms. Probably that behavior implies some function here that we are not entirely sure what it is. It looks like mostly the sodium go in and out through the triangular pores that are very electronegative around or their periphery. It might relate to, I used to like to think that this had to do with exposure of that arginine-rich domain, and it was an electrostatic thing, but now we have the cryo-EM reconstructions with it coming out of the 6-fold instead, so I don't know, we're going to have to wait to find out what that does functionally.
Jodi, great talk. I had a question about your first part where you see the peeling of the tips. How often do you see that in your simulations, and is there any experimental evidence for it?
Yeah, so this thing, I want to say there's maybe 4 copies at a time doing it in the capsid, but they don't stay open for the entire simulation. I keep asking my student to tell me exactly how transient it is. We don't have that yet. So it's not like all the spikes are doing it all the time. It's relatively rare, which is why you sort of just get that shoulder in the distribution, and you don't even see it in the apiform capsid in the distribution, even though it's doing that. Just there's not enough copies. Yeah, and in terms of an experiment, I would really like one of my experimental collaborators to maybe engineer a crosslink right there for me, and maybe we do some experiments, we see how that affects assembly. Maybe it doesn't assemble anymore, and we see that this is assembly inactive, for example. Does it make capsids? Do the capsids, export through the nuclear envelope faster? Maybe we could start to find out what this conformation is good for. But if you have some ideas, please let me know.
Hi, Jodi. Very nice talk. Do you see any correlated motion between the capsomers, or is everything independent from one another?
And there's a lot of correlated motion. There's a lot of really messy correlated motion because we've only run for a microsecond. It's this big simulation, 6 months, we pat ourselves on the back, but at a global level, the data set is unconverged. So there's a lot of correlated motion. It's very difficult to interpret because it's asymmetric, and literally you do a principal component analysis and look at some of these correlated motions, and you can see everything going on at once, all the different motions. It's really hard to make some sense of it. We've almost got worked out now a way to symmetrize these modes where we don't introduce artifacts, so we can maybe interpret what we think what a symmetric mode that includes really long sampling time, a converged data set, what this symmetric breathing modes might look like. So maybe next year I can show that.
Okay, last question, Paul.
So with the opening and closing of the dimer, has anybody made any mutants on that asparagine or arginine pair, including in silico mutagenesis, to see whether or not it stays in position or for some other conformation? Because it's really interesting. That's quite a long time that it's open and quite a long time that it's closed. That's really a cool thing.
I haven't seen experiments like that, and we haven't been able to convince a collaborator to do it yet for us. I think it's pretty, one comment I can make is that this appears to be quite conserved across, if we do sequence alignment with many different strains. One time we did see the arginine substituted for a lysine, so, maybe that makes some sense. We got really interested in this salt bridge in the paper that we published this in because we were really looking for allostery in the capsid and how the CAM molecule might be inducing, things to happen elsewhere in the capsid. So, this integrity of the intradimer interface should be important for assembly. Assembly happens out here, it's pointed out at you, in the interdimer interface. So how do these two domains talk to each other? People have often said that, well, it just travels down and out, but what we show is that if we look at correlated motions in the dimer, that this helix that's sticking out towards you, this is our helix 5, our interdimer interface that contacts the next dimer. It's dynamically linked with this fulcrum. These guys are working together, and now you have a direct contact between the spike tip and the fulcrum, which is, again, working with the interdimer interface. So this could be a previously unproposed pathway for allosteric communication rather than sort of just going down and out. So maybe.
Yeah, thank you very much, and let's thank again all the speakers of this session. And now it's dinner time, you know where to go, and Fred just reminded me, and I think it's also clear that we have afterwards, from 20 or from 8 o'clock, we have posters, and enough time to discuss all this nice work on the posters. Thank you.